This is Dr. A. Gokulakrishnan presenting patterns of inheritance. There are two different types of inheritance. One is a Mendelian inheritance and non-Mendelian inheritance. Now, first we will look into after Mendelian inheritance. Genetics is a science of heredity. It is the branch of science dealing with the way in which genes are inherited from the parents to the offsprings. Gregor John Mendel is often called father of genetics because he was the first person to discover how traits are passed from parents to the offsprings. In the late 1800s, Gregor John Mendel, an Austrian monk and a plant breeder, conducted experiments which lead to discovery of genetics. This figure shows the homologous chromosomes and sister chromatids. The expressible part of a DNA, it is called as gene. First, let us introduce few terms. Mendelian factors are now called as gene. That is, expressible part of a DNA, it is called as gene. Alleles are different version of the same gene. For example, a tall and dwarf gene, blue eyes, brown eyes, and green eyes. Dominant allele, a trait that exerts its effect whenever present, it is called as dominant allele. It is symbolized by a capital letter for capital letter. The recessive allele, the trait that is masked if a dominant trait is present, it is symbolized by a lowercase letter. Genotypes refer to the genetic composition of an individual. An individual with two identical genes, it is termed as homozygous. For example, tall gene, which are capital T and capital T. An individual with two different genes is termed as heterozygous. Heterozygous tall, it consists of a capital T and small t genes. Phenotype refers to the outward appearance of an individual. Example, blue-eyed, brown-eyed, tall or dwarf. Generation. A group of organisms born around the same time, it is called as generation. There are three different types of generation. One is parental generation, that is P generation. F1, first filial generation. F2, the first generation offsprings. First, we will look into after Mendel's monohybrid cross experiment. The first experiment of Mendel considering the inheritance of single trait, that is height of a plant, a tall or dwarf, is called as monohybrid cross. For his monohybrid cross experiment, he selected a pure homozygous tall and dwarf garden pea plant, that is Pisum sativum plants. He crossed a tall plant with a dwarf plant and observed how the traits are transmitted the progeny, and calculated the percentage of tallness and dwarfness in subsequent generation. When a pure breeding tall plant was crossed with a pure breeding dwarf plant, all the plants were tall in the first filial generation. When such F1 tall plants are allowed to self-pollinated, both the tall and dwarf ones were appeared in the second generation. In modern terms, the homozygous parents in the P generation can pass one kind of allele to their offsprings. For example, a homozygous tall plant can pass only a tall gametes, possess tall gametes. The heterozygous parents of F1 generation have two alleles and can pass one or the other but not both to their offsprings. This is the diagrammatic representation of monohybrid cross. It took a homozygous tall plant and it crossed it with a homozygous dwarf plant. In the F1 generation, all the plants appeared to be tall. Their phenotype is tall while the genotype is heterozygous. Now he self-pollinated this F1 hybrids, two F1 hybrids. He produced 4 different plants: a tall plant, homozygous tall plant, and 2 heterozygous tall plant, and 1 homozygous dwarf plant. In the F2 generation, the genotypic ratio is 1:2:1, while the phenotypic ratio is 3:1. A Punnett square is a grid that enables one to predict the outcome of simple genetic crosses. It was proposed by an English genetist, Reginald Punnett. We illustrate the Punnett square approach using the cross of heterozygous tall plant as an example. Write down the genotypes of both the parents. A male parent in the F1 generation possesses a capital T and small t, while the female parent also possesses a capital T and small t. Write down the possible gametes of each parent can make. The male gamete possesses either a capital T or a small t. A female gametes also possess either capital T or small t. Create an empty Punnett square and fill up the male gametes and female gametes and cross it to produce the offsprings. Determine the relative proportion of genotype and phenotypes of the offsprings. The genotype is 1:2:1, while the phenotypic ratio is 3:1. Here, this is the Punnett square of both F1 generation and F2 generation. In the F1 generation, all the plants are heterozygous, while in the F2 generation, there are 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. Based on the monohybrid experiment, he proposed a law of dominance. A pea plant contains 2 discrete hereditary factors, one from each parent. The 2 factors may be identical or different. When the 2 factors of a single trait are different, one is dominant. Its effect can be seen. The other is recessive. It is not expressed. It is hidden. The law of segregation. During gamete formation, the parent factors segregate randomly so that half of the gametes received one factor and half of the gamete received the other. This law, it is called as Mendel's law of segregation. Backcross, a cross between F1 hybrid and one of the homozygous parent. It is called as backcross. There are 2 possibilities: the cross of hybrid with a dominant parent and a cross of hybrid with a recessive parent. When it is crossed with a dominant parent, it results in 100% tall plants. When it is crossed with recessive parents, a 50% of tall and 50% of dwarf will appear. So this second cross, that is a cross between a heterozygous plant and the homozygous recessive plant, it is called as test cross. Back cross is very significant as it can be helpful to the farmer or the plant breeder to design the breeding as per the demand. For example, if purple flowers are more in demand, then the breeder should try to cross F1 hybrid with the dominant parent. If the white flowers are also in demand, then the breeder should cross F1 hybrid with the recessive parents. Mendel's dihybrid cross experiment. Mendel also performed a dihybrid cross, crossing an individual plant with different 2 traits. For example, the trait 1 is seed structure, a round seed and a wrinkled seed. Trait 2 is seed color, yellow color seed and a green color seed. There are 2 possible patterns of inheritance. It may be either a linked assortment or an independent assortment. Mendel want to test whether it is an independent assortment or a linked assortment. First, he took a round yellow plant and crossed with a green wrinkled plant. In the F1 generation, he got only a round yellow plant. All the plants are round yellow. Then he self-pollinated the F1 hybrid. It produces 16 different plants or 16 different plants. Of these 16 different plants, 9 of them are round yellow and 3 of them are wrinkled yellow and 3 of them are round green and 1 is wrinkled green. Data from hybrid cross. When Mendel crossed a round yellow seed plant with a wrinkled green seed plant, in the F1 generation, all the plants are round yellow. In the F2 generation, he got 315 round yellow seed plant and 101 wrinkled yellow seed plants and 108 green round seed plants and 32 green wrinkled seed plants. If the genes assort independently, then the predicted phenotypic ratios in the F2 generation would be 9:3:3:1. The Mendel data was very close to segregation expectation. Thus, he proposed the law of independent assortment. Based on the Mendel's dihybrid cross experiment, he proposed two laws. One is law of segregation. The two alleles of each gene are packed into a separate gametes. The allele move apart during meiosis. The next is the law of independent assortment. The segregation of alleles of one gene does not influence the allele of the for the another. So the alleles assort independently. The alleles are randomly packed into the gametes. Now we will look into the probability. It can be used to better understand of segregation and independent assortment. First, look at a tossing of a coin. If you toss a coin, what is the chance of head to get an head? Half a chance are there, that is 0.5. And what is the chance of a tail? Half a chance are there, that is 0.5. Based on this, there is a probability rule, the sum rule. The combined probability of two or more mutually exclusive events occurring is equal to the sum of their individual probability. For example, a parent has a capital G and small g genotype, that is, it is heterozygous. What is the probability of the offsprings which will receive a capital G or small g allele from the parent? The probability of capital G or small g is equal to 0.5 plus 0.5, that is equal to 1. So the combined probability of two or more mutually exclusive events should be equal to the sum of their individual probabilities. Then product rule. The joint probability that both of two independent events occur is the product of an individual probability of each event. A mother has a capital G small g genotype and father has a capital G small g genotype. What is the probability that the offsprings will have small g genotype? The probability of G small g from the mom and and small g from the dad is 0.5 into 0.5. The probability of small g genotype in the offspring is 0.25. So the joint probability that both of the two independent events occur is the product of individual probability of each event. Now we will go into autosomal dominant disorder. The dominant disorder is a condition is expressed when an individual has at least one dominant allele, diseased allele. For example, an Alzheimer's disease. Even in the heterozygous condition, it is affected. Recessive disorder, autosomal recessive disorder. Example, sickle cell anemia. Due to the presence of a recessive allele, it is denoted by HBS. It causes the distortion of red blood cells into long and thick sickles. It causes pains and tissue damage and cells to rupture prematurely. The heterozygous are carriers and do not manifest the symptoms. Only the homozygous are the diseased one. Homozygous recessives are the diseased one. Punnett square, it is used to predict the possible offsprings of the genotype. Place the alleles for each parent side. For example, the mother genotype, it is normal HbA, while the father genotype, it is an sickled cell, that is HbS. Then there are 4 different types of offsprings. All the offsprings are heterozygous, and are normal. Okay, the heterozygous offsprings are carriers. Here is the list of some of the autosomal recessive disorder and dominant disorders. Now we will go into the second part, the non-Mendelian genetics. The non-Mendelian genetics describes the inheritance pattern based on complete dominance or recessiveness. There are other types of inheritance that is Mendel never considered. It is incomplete dominance, codominance, multiple alleles, polygenic traits, and sex-linked inheritance. In some cases, one allele will not completely dominate over the other allele. That results in a blending of two alleles. For example, a red snapdragon is crossed with a white snapdragon. The resulting offspring will be a pink in color instead of getting a red due to the incomplete dominance. Here, there is a problem related to incomplete dominance. What is the probability of pink color flowers if pink flowers are breeded with red flowers? When the pink flowers containing plants are breeded with red color flower, 50% chances are therefore to get a pink flowers. Codominance. Both the traits are dominant and show up their phenotype together. Co means together. In some cases, both alleles are dominant and expressed at the same time. They are called as codominant. Generally, it results in a spotted appearance. For example, a black cow, when it is crossed with a white cow, here we will get a spotted cow. That is, in a white cow, there will be black spots. This is due to the codominance. Both the white and black appears side by side. Here is the problem related to codominance. What are all the possible phenotypes when two spotted cows are breeded? When two spotted cows are breeded, the possible phenotypes are black cow, two spotted cow, and a white cow. The human blood type displays both codominance and complete dominance. The red blood cell can either have a carbohydrate moiety on their surface or not. The presence of carbohydrate, that is high, is dominant in the absence of carbohydrate, small I. Additionally, there are two different carbohydrates that may exist on the surface of RBC called as IA and IB gene, group A and group B respectively. The cell surface carbohydrate A and B are codominant, which means they could also show up at the same time on an RBC. Based on this, there are four different types of blood groups: A, B, AB and O. Here there is a problem. If a woman with a AB blood has a children with a man who has O type, what will be the possible genotype of their children and what will be their blood types? All of them are either 50% of them are A group and 50% of them are B. That is, they are heterozygous in nature. Multiple alleles. When more than two varieties exist, multiple alleles. In some cases, more than just two alleles exist in a population. The gene is to have a multiple allele. Multiple alleles can follow any type of inheritance that we have talked about. Example, blood types IA, IB, then small I, type O blood in a homozygous state, and IA and IB are codominant to each other and dominant to small I. Many animals have a variety of coat colors. This is due to the multiple alleles. Now we are going to see about polygenic traits. Require more than one gene to determine the trait. Example, human skin color, eye color and height. Skin tone is determined almost 6 genes. That means there are 6 different chromosomes are involved. Now we are going to see about sex-linked triads. A sex chromosomes are referred to as X or Y. The male contains XY while the female contains XX. Some genes are only present on one of the sex chromosome, usually X. The female with recessive allele but dominant phenotype. The male with recessive allele and recessive phenotype. Example, color blindness, male pattern baldness. The genes for some traits are found on the sex chromosome XY. Most of the traits are recessive and the normal gene is dominant. Heterozygous female are carriers. They do not show the trait but they carry the gene for the trait. Homozygous female have the trait. The males with the gene XY have the trait. They do not have another X to counterbalance the affected genes. Hemophilia is a rare hereditary human disease of the blood in which the blood does not clot properly. A normal man carries hemophilia woman. A normal man marries hemophilia woman. They have a son with hemophilia. Can they have a daughter with hemophilia? Show your work using a Punnett square. Here this is the Punnett square of a hemophilia female with a normal male. When they get married, they will produce a hemophilia sons, while the daughters are carriers since they are heterozygous. Color blindness is a sex-linked recessive trait. The gene for this trait is inherited through the X chromosome. For example, if a woman with a normal vision marries a man who is colorblind and has a children with a normal vision, what are the chances their children will be colorblind? From the Punnett square, we come to know that all the childs are normal. Will any child be carriers of the trait? Yes, the female childs are the carriers. Thank you.